Hello once again. So here we are back with chapter 12, which is on gene mutations. Now last time we looked at sections 1, 2, and 3, and that was just kind of a basic understanding of what mutations are. We talked about 2 beta globin gene mutations, so mutations in the beta subunit of hemoglobin, and those were sickle cell disease and beta thalassemia. We talked about 2 types of collagen disorder. So collagen is a protein that allows for flexibility and strength among your different tissues. We talked about Alport syndrome and Marfan syndrome. And then we even mentioned an allelic disorder, which was in the presenilin protein disorder, which led to early onset Alzheimer's. Well, today we're going to move on to sections 4 and 5. So 4, we're going to talk a little bit about why mutations arise. And 5, we're going to talk about actual types of mutations. So let's get ready to rumble, shall we? All right. Oops, wrong button. Way to go, Komar. There we go. There we go. So, why do mutations arise? Remember, we know that they are changes to the actual DNA sequence. So it's a change in the bases, whether it's the order of the bases, which exact bases are present, how many bases are present. But they can actually arise from many different ways. The most common is just an error done in DNA replication. We replicate our DNA frequently, especially with those cells that divide frequently, like our skin cells. So it can be pretty easy for an error to occur. Let me move this, sorry, this is bugging me now. There we go. So it can be pretty easy for errors to occur when you replicate your DNA so often. However, it doesn't mean that's the only way they arise, and we are going to talk about a few other things involved with mutations coming up. Oh, that's right. So first we're going to just mention something called Talomerism. Try saying that 5 times fast. So this is a particular spontaneous mutation, and what it is it occurs when there are rare tautomers of bases that are actually incorporated. So this is one that occurs during DNA replication, but it makes these rare forms of the of specific bases. Now you remember adenine and thymine can form 3 hydrogen bonds, guanine and cytosine can form 2. But what happens is these tautomers, it's a rare form of that particular base, and it actually changes the number of hydrogen bonds that it can make. So tautomerism, a base is changed by the repositioning of a hydrogen atom, altering the hydrogen bonding pattern of the base, resulting in incorrect base pairing during replication. So if we look here we have thymine, and normally that should form hydrogen bonds with adenine. But here we have a rare form of guanine. It's a tautomer of guanine. And it can form 3 hydrogen bonds, which lets it bond with thymine. Now, normally it can't do that, but because of a hydrogen atom being rearranged, and it allows it to form a different number of hydrogen bonds, thus allowing it to bond with its typically non-complementary buddy. mDNA mutations. So mDNA is mitochondrial DNA. Now the mitochondrial DNA can actually replicate, it can actually have an increased rate of mutations occurring in them, simply because the mitochondrial DNA does not have any repair mechanism. Later on, we're going to, in section 6, we're going to talk about some ways that our DNA can actually repair itself if it finds a mutation. But there are no repair mechanisms in the mitochondrial DNA. So therefore, if a mutation occurs, then we tend to see these more often than in your nuclear DNA. So back in the day, there was a Chernobyl nuclear accident. And what they've noticed is that it actually did have some effects. Even though people were rushed out of the area, they were still exposed to certain levels of the radiation. And it may not have affected them a whole lot, but what was noticed is that it affected the DNA in the mitochondria of the eggs or sperm. So gametic mitochondria and gametic cells. As a result, when these mutated gametes fused during fertilization, they saw babies born with different muscular disorders. Because remember, the mitochondria, it makes energy for the cell. And specifically, there's a lot of mitochondria in the muscle cells because, well, they need lots of energy. As you can tell with this child here in the picture, you can just see that the muscles are not formed correctly, and a lot of it is because of these mutations in the mitochondrial DNA. All right. Bacteria and viruses. Yeah. So they actually get lots of mutations. This is why we like to study them in terms of actually also adaptations and evolution, because since they reproduce so frequently, they actually have a very high spontaneous mutation rate. What that means is, for us humans, one generation can take, 25-ish years, give or take, maybe like 20 years on average. Well, one generation for a bacterial cell or a viral cell is a few hours. So that means in the span of a day, you can have multiple generations of these born. In the span of one day, you cannot have multiple generations of humans. In the span of a decade, you really can't. So because they reproduce so quickly, it allows us to see mutations occurring more readily. So they can get a much higher rate of mutations. And like I said, that's why we like to study them, especially when it comes to like antibiotic resistant bacteria. It helps us to study them because many of them start to mutate and become immune to our antibiotic medicines. Now, that means that they're more likely to have a higher rate of mutations. And sometimes when they infect their host cell, they can actually leave a part of their own genome within the host genome. We've mentioned previously how there is actually viral DNA within our own genome caused by an ancient ancestral infection. So there is viral DNA already in our genome. It wasn't there originally. It was there when there was this ancient infection, and it became part of our DNA. Now, if there's a mutation in that bacteria or virus DNA, and now it's part of our DNA, well, that's a change to our DNA, and that is a form of mutation, or a way that mutations can arise. Induced mutations. Sometimes we want to study how different genes work. So they do this in a lab for research. They're not just going to go and mutate people. Sorry, no, you cannot go to your doctor, ask them to induce a mutation and give you superpowers. How this works. But scientists can actually purposely cause mutations in cells in order to study them. Now it says researchers use mutagen chemicals, so chemicals that can cause mutations, and radiation to reveal normal gene function. So we know that we have a whole bunch of different genes in our cells or in our DNA. We don't always know what their function is on their own. So what they might do is take a cell or a colony of cells, measure what kind of proteins are being made by that cell. They can then induce a mutation and see if that changes the proteins that are being made structurally or the amount or anything like that, in order to figure out, okay, if it makes this protein, maybe it has this function. Ionizing radiation can also be a way that they induce mutations. The reason why this works is because it actually breaks the sugar phosphate bonds in DNA. However, that's not going to change the bases. That's going to break sugar phosphate backbone and can actually result in the DNA falling apart. And they typically do this in the form of gamma rays. So no, do not go trying to shoot yourself with gamma rays to become the Incredible Hulk. Bruce Banner was lucky, and that's fiction. Can also do site-directed mutagenesis with PCR. So we've talked about PCR in the past. That's polymerase chain reaction. And that allows us to make many copies of one gene. So what we can do is mutagenesis just means causing a mutation. We can cause a particular mutation in that gene that we are making multiple copies of, again, in order to research to see what kind of effect it could have. All righty. Hotspots. So a hotspot is simply a section of the DNA that is more likely to have a mutation occur in it. So mutations are more likely in or near repetitive or symmetrical sequences. Now, we've talked about signs and lines, how they are. Signs are these short repetitive sequences, lines are these long repetitive sequences, and I've even told you that it's believed that they're used as buffer zones. Because of the nature of repeating sequences, there's more likely to be an error in that section of the DNA. And if you have .200 bases long for your DNA, it's much bigger than that, then that means 4 of those are going to code for your DNA, and the other 196 are not. They do not code for your proteins. 4 do, 196 do not. Within that 196, you have repeated sequences. Well, that's where the signs and lines are. And it's these repeated sequences that make up these large chunks that actually provide buffers by being more likely, it's a game of numbers, you're more likely to have a mutation in this block of 196 bases than in this block of 4. So it allows for the possibility of mutations occurring there rather than in a gene that can have some sort of important function. All right, natural exposure. So there are certain mutagens that naturally occur around us. So there is radon from the Earth's crust. It is so minute, you don't have to worry about it. But that is why there's thought that if you prolonged excursions underground, like deeper underground where there's more radon, could potentially lead to certain mutations or effects. Sunlight, we know that can lead to skin cancer, and that's caused by a mutation in your DNA. But just like how being under the ground may or may not cause an increase in radon exposure, being up in space is one reason why they have to, why astronauts can only spend so much time in space. I mean, there's a whole loss of bone density because there's no gravity, so your muscles don't have to rely on your skeleton as much. But also, you are outside of the Earth's atmosphere, and the Earth's atmosphere blocks a large chunk of radiation from the sun. So they can be easily exposed to many more times of solar radiation than we are. So that's why they have to be very careful about how long they spend in space at a time. Yeah, wrong button, wrong button. All right, let's move on. There's something called the Ames test. And it's a type of test that's used to look for the rate of the mutation. So how quickly can a mutation occur? So the method that uses bacteria to test whether a given chemical can cause mutations in the DNA of a test organism. So, sorry. So biological assay to assess mutagenic potential of chemical compounds. So what we mean by that, here, we have a strain of salmonella, and it comes from rat liver extract as the base solution, and then the bacteria salmonella is in there. And what we're going to do is we're actually going to separate into two groups. We are going to use one as just kind of a control. So we're just going to take that as is, plate it in a petri dish, incubate it to see how much bacteria can actually survive. Now, in this cause, specifically salmonella needs histidine. There's not a whole lot of that in rat liver extract, which is why you don't see a whole lot of them growing. But what we're also going to do in the other group, we're going to add a possible mutagen to them. So something that may or may not have an effect on their DNA that causes them to change some gene. Then we're going to plate them. And if you notice with both of them, the agar has minimum histidine, which is why a little bit can grow here. But this one, still just a minimal histidine in the agar. But now we also have the possible mutagen. We incubate it. We notice that there's a lot more of the bacterial colonies, of the salmonella colonies. So what that means is that this mutagen allowed the salmonella to now thrive in the same environment, but with greater success. So a high number of revertants suggests a mutagen causes mutation. So we can see that the fact that so many of them were able to survive and have colonies meant that there's probably some mutation that occurred that allowed them to survive in this environment, whereas not that many survived here. So that's just what an Ames test is. You induce a mutation or add a mutagen to bacteria in order to figure out if it has an effect. Does it cause a change? If we saw the same results for both, then that means that this possible mutagen does not cause a mutation. But since we see a clear change, it means that it did. All right, let's talk about types of mutations. First one, we have these things called point mutations. Now, a point mutation is when there is a change to one single base, just one. So we have a change to a base. Now, sometimes there's zero effect. So it just kind of results in the same amino acid. So if we look down here at the picture, here is the original DNA, TTC. It makes that particular, that particular mRNA, AAG, and that codon codes for lysine. Well, a silent mutation is when there's a change. So we do see a change. This cytosine is exchanged and substituted for a thymine. So we went from TTC to TTT. That changes the codon, but it does not change the amino acid. We still get lysine. So that's what we mean by silent mutation. There's a change to the DNA, but no change to the amino acid. We can then classify the other point mutations as transition versus transversion, or missense versus nonsense, which we'll talk about both of those right now. So transition versus transversion, this deals with what kind of base is being exchanged for what kind. So in a transition, a purine replaces a purine. If you remember correctly, adenine is a purine, and so is guanine. So what happens is you can exchange an adenine for a guanine or a guanine for an adenine. So you're exchanging the larger purine for another large purine. Or A pyrimidine replaces a pyrimidine. So thymine and cytosine are the pyrimidines. They're smaller. They're a single ring structure. If you originally had a thymine and it gets exchanged for a cytosine, that's a transition as well. So you're taking a like base and exchanging it for a like base. So purine for purine or pyrimidine for pyrimidine. Transversion is when you are switching unlike bases. So you're starting off with a purine and replacing it with a pyrimidine. So maybe you're starting off with adenine and replacing it with thymine or cytosine, or guanine, replacing it with thymine or cytosine, or vice versa. So you can also start off with a pyrimidine, thymine, and replace it with a purine like adenine or guanine. So transition, like for like, purine for purine, pyrimidine for pyrimidine. Transversion, different. Purine for pyrimidine or vice versa. And like I said, we can also classify them as missense versus nonsense. And that's more based on the effect it would have on the amino acid chain. So a missense is when you exchange one amino acid for a different one, like we saw with sickle cell anemia, how in the original DNA it was glutamic acid, but in the sickle cell mutation, the glutamic acid is exchanged for valine. If you look down here, right here, so TCC as opposed to TTC. So thymine, thymine, cytosine became thymine, cytosine, cytosine. That changed the mRNA and exchanged the lysine for arginine. Now that's an exchange of a pyrimidine for a pyrimidine. So not only is this a missense, it's a transition missense. Or we can have TGC. So again, TTC to TGC, that affects the mRNA codon, which is going to cause a different amino acid, threonine. So lysine is exchanged for threonine. And not only is this a missense because we exchanged one amino acid for another, but we are exchanging this pyrimidine T for a purine, guanine. So that's a transversion missense. And then we have nonsense mutations. So this changes the amino acid code to a stop codon. And what that means is if we have a stop codon in the middle of our gene, then it's actually going to halt translation at that point, and none of the amino acids beyond that point will get added. So that's right here. So we exchange the thymine for adenine. Hey, look, that's a transversion. And that changes the mRNA to UAG, which then it leads to a stop codon. So any amino acids beyond that point will not get added on. An example of this is Becker muscular dystrophy. And what happens is that the protein is actually shortened by 15%. So the first 85% of the gene is translated, but then they have a mutation that causes a stop codon too soon. And that last 15% doesn't get translated because of that early stop codon. All right. I'm going to talk about splice site mutation. So splice site mutation is when there's an alteration of the site where introns are normally removed, and it may cause exons to be skipped. So this will make more sense once we look at the picture. An example is familial dysautonomia, or FD, because that's quite a mouthful. Now, what this leads to, what are some of the symptoms of FD are frequent vomiting, pneumonia, problems with speech and movement, difficulty swallowing, inappropriate perception of heat, pain, and taste, as well as unstable blood pressure. Now, that inappropriate perception of heat, pain, and taste, think of it like this. If you were to touch the top of a stove while it's on, how would that feel to you? It would feel hot, right? Hot and painful. It would cause you to jerk your hand away. But what if it felt like a slight tickle instead? You may not move your hand away even though it's hot and it's burning your skin, but you're not interpreting that perception as heat or even as pain. And so you leave it on there longer, and now you got third-degree burns on your hand. Or taste. We can agree that sugar tastes sweet. But what if it tastes like fish? Imagine that. So it's these changes in how different neurological sensations are being processed. How are you interpreting the sensation of heat? How are you interpreting the sensation of pain? How are you interpreting the sensation of taste? So there's a lot of different things with that. All right, so here we have exon 11, 12, and 13. Now, typically what happens is these introns get cut out and we have 11, 12, 13. So they get put together, we have a correctly made protein. But what can also happen, the splice site, is that, notice there's a guanine here in the original one. So what's going on is we are splicing out from this region to this region. So it's going from adenine to guanine. Now, if this guanine gets changed into an adenine, it's going to be completely passed over until the next guanine section, the next GU section. So it's that GU that we're looking for. So what happens is this AAG starts it. So AAG starts it. This adenine gets cut all the way down to where there's a GU. So that part gets cut out, removed, keeps reading. Well, we got our A and G, start cutting from this adenine all the way down to GU. But if this is no longer a GU section, then it doesn't get recognized, which means that when we start splicing and cutting out that intron, this entire section gets cut out until we finally reach a GU. Now that means that we're losing that exon. So now we just have 11 and 13. So we are missing that whole exon, that whole section of bases for this protein. All right, frameshift. So frameshift is also known as insertions or deletions. And the reason why we call them frameshift is because they shift the reading frame of the gene. Now, what we mean by the reading frame is how we look at the codons in order to figure out the amino acids. And think of it if I have 20 kids lined up in a row. If I take 2 of the kids from the middle out, well, the rest of the kids are gonna move forward to fill in that gap. Or if I take kids from another classroom and stick them in the middle of the line, everyone else will have to move backwards to make room for them. And that's what's going on in this case. Now, if it's an entire codon that is inserted or deleted, it actually won't cause a frameshift mutation, because at that point you're adding a codon, which means you are adding a new amino acid, or you are taking out a codon, so you are taking out an amino acid. But the rest of the amino acids in the back are left unchanged. But if you only add 1 or 2, then since that shifts all the letters down, it's actually going to alter every single amino acid beyond that point. So we see big changes with frameshift mutations. And it can be as little as 1 base being added or deleted, or thousands of them being added or deleted. So some examples of it is male infertility can be caused by deletion on the Y chromosome. So there's actually a section of one of the genes on the Y chromosome that if it's missing, you still have a developed male, but they cannot, their sperm essentially cannot fertilize any eggs. Let's see. So 2/3 of people with Duchenne muscular dystrophy are missing huge sections of a particular gene. Now, if you remember, Duchenne muscular dystrophy is sex-linked, so it's located on the X chromosome. And what they're finding is that many people with Duchenne, one of the reasons, or one of the things that causes the symptoms, is that there are sections of the gene missing, like these rather large sections. And because of those sections being missing, it affects the proteins, which then leads to the symptoms of Duchenne. Gaucher. So that's actually named after a French guy, so it's Gaucher. And there's an inserted single base in DNA. And what this causes is a short or non-functional gene. And the reason why it causes a short or non-functional gene is because by adding that base in, it could actually lead to a very early stop codon. So that means that not as much is being made. So that could be why it's short. And since it's so short, not a whole lot, there's not enough of it to do any job. So it results in it being non-functional. So here's an example of what a frameshift would look like. I'd also recommend checking out my Science in Pajamas frameshift mutation video. I do a whole bunch on the board to kind of demonstrate how that would look. But anyways, normal DNA. So here we have 1, 2, 3, 4. We have 4 codons and an extra A. So we go through, we transcribe it. Here's our mRNA. We translate it. We get leucine, alanine, cytidine, and isoleucine. Yeah, I think that's what that one is. But here we have an addition of a guanine between the T and the C. Remember, they used to be next to each other right here. Well, now that pushes all of these back. So instead of GAT, okay, we still have that. Next one should be CGT. Well, because of that G being inserted, it's no longer CGT, it is GCG. Then the next codon is TAC. Then the next codon is ATA. And now we have a T and an A hanging off the end. So it still gives us leucine in the front, but then we get arginine, methionine, tyrosine. So you can see how that one insertion pushes all the bases back, which is going to cause all the codons to shift, which means that we're going to have changes in the amino acids all the amino acids after that point. All right, and the last thing we're going to talk about is other types of mutations. So we're just going to kind of talk about them very briefly. First one is called Tandem Duplications. So this is, again, we're just going to kind of mention these. We're not going to get into a whole lot of detail. Tandem bicycle is when you have 2 seats, so 2 people sharing one bike. So this is when you have 2 copies of a gene right next to each other. So notice in the original chromosome, we have genes A, B, C, D, E, F, G. Well, here in the tandem duplication, we have A, B, C, D, E, F, G. So again, we can see how there are 2 copies of this section. So we have this section here, and we have another section of it as well. Pseudogenes, we've talked about these. They are essentially non-functional sequences. They are similar to nearby genes though, and they may get transcribed, but they often do not get translated. However, since they are very similar and very close to other genes, what happens is they can actually cause a disruption during meiotic pairing. And what we mean by that is they can actually cause a crossing over event between the functional gene on one of the homologous chromosomes and the pseudogene on another. So if you look down here, let's see. We have the gene, goes through duplication and mutation. We get duplicated pseudogene, which means we actually now process them. Or we can transcribe the gene, process it, and we get our mRNA. So that can then lead to, and pictures a little weird to really look at, but we think about it this way. You have the homologous pairs. There's my fingers. Now they should cross over. So the top part of my finger should cross over, and we end up with 2 fingers still with all their 3 sections, each of the, 1, 2, 3, 1, 2, 3. But what if this top section of this finger and this middle section of this finger switch places? Well, now it's top, top, bottom, and it's middle, middle, top, bottom. So that's what we're kind of looking at. And again, Gaucher disease, it results from the crossing over between the actual gene and its pseudogene. Transposons, these are also known as jumping genes, because what happens is they can actually remove themselves from the part of the DNA and insert themselves somewhere else. So it can, specifically, they can cause disruption if they insert back into the DNA in the middle of a gene. Now, if you look at here, here is the chromosome as is. We have the transposon here. It cuts itself off and it inserts itself in the middle of the gene. Well, now when that gene gets transcribed, you're going to have this whole extra section in there, and that can affect the function of the protein. Expanding triplet repeats. So expanding triplet repeats lead to protein misfolding, and it will worsen with each generation. And the reason why it worsens each generation is because with each generation, that triplet repeating section, you get more of it. That's why we call it expanding triplet repeats. It's kind of like the opposite of telomeres. With telomeres, they're the ends of the chromosomes. They shorten every time the DNA gets replicated. But in this case, this section of triplet repeats, now triplet, we just mean 3 bases. So let's say ATG. So we start off maybe with 50 copies of ATG. Then the next generation, maybe there's 75 copies of ATG. And the next generation, 150 copies. And the next generation, 275 copies of ATG. Each generation, we're seeing more and more of this repeating section, and that's why we call it expanding triplets. So let's see. An extra amino acid may impart a new function to the expanded protein, because since we are expanding it, that means that there's going to be more codons in there, which means more amino acids, and that can affect the function of the protein and can often disrupt brain function or affect export or translation of the mRNA. So there's something called Fragile X syndrome, and that's what we see in the 2 guys here on the right-hand side. Now, normally, in a normal chromosome, here's the X chromosome. At the bottom here, that is the FMR1 gene. But in Fragile X syndrome, what happens is there's a mutation, a triple, or this extended triplet repeating section that occurs in the middle of the FMR1 gene. So it gets this really, really thin section of these repeating sets of triple bases, and it actually pushes it out. Now you notice there's a bit of a gap there, and that's why they call it Fragile X, because those ends could actually be broken off and you would lose the genes at the end of them. So Fragile X syndrome is, they've actually found that it only occurs in males. Meiotic dystrophy or even Huntington's disease is a result of these expanding triplets. So an example of one, which also leads to this, normally there's about anywhere from 5 to 37 copies of CTG at a particular location in the X chromosome. That's normal. But what they found is when there's 50 to 1,000, you start seeing some of these different disorders, again, depending on the exact location of this expanding triplet. And that's it for this one. We finished we finished the second video presentation, sections 4 and 5. If you have any questions, hit me up in our Google Classroom. But until then, I will talk to you later. Oh yeah, or you can email me. But take care, you guys. Hope all is well, stay healthy, and just stay awesome. All right, you guys. You take care.